Hello everyone, and welcome to Designing the Ideal Multiparticulate Formulation for Pharmaceutical Use presented by Capsugel. I am Amira Al-Idris, editor for the Fierce Markets Life Sciences Group, and I'll be moderating this webinar. Our speaker today is Dr. Brett Caldwell, Director of Analytical and Quality Control, Bend Research. You can read his full bio on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to participate in the interactive parts of this presentation. If you'd like to download the slide deck, please click the Download Slides widget at the bottom of your screen. This webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentation by a Q&A session. Please submit your questions during or after the presentation using the Q&A box at the right of your window. Now let's begin. Brett, please go ahead.
Hello everyone, my name is Brett Caldwell. It is a pleasure to be able to spend an hour today talking about one of my favorite formulation topics, the immense capability and flexibility multiparticulates offer a formulation team to achieve its goals. Let's face it, all new chemical entity or 505(b)(2) formulation teams want a differentiated formulation that will make the boss smile. The patient, consumer, and company benefits from such a differentiated formulation can arise from achieving goals such as robust formulations for new chemical entities, improved efficacy or safety profiles for existing pharmaceutical ingredients, adapting from immediate to modified release, which yields a lower pill burden and better patient compliance, and even better processes that lead to lower costs for our medications. In the course of achieving those goals, we all want the ability to design, develop, and scale up to clinical and commercial manufacturing as cheaply or rapidly as possible. Commonly, that would mean a team chose right the first time the formulation for the potential product and efficiently got it made and tested. My main hypothesis for today is that the precedence of multiparticulates in the market and the maturity of the processes for making them should really result in multiparticulates being a primary formulation tool for almost every formulation team. Secondly, the selection of an optimal multiparticulate technology for any given product should be primarily focused on meeting pharmacokinetic and pharmacodynamic targets, being accompanied by a fundamental understanding of each multiparticulate formulation and process type available to speed the development along the desired path. With those as my premises for today, it must mean multiparticulates can be used to achieve almost any target product profile goal a team might have. Market research indicates that multiparticulates are playing a large role in life cycle management strategies and submissions to the FDA. A primary reason for this growth is the ability to use various multiparticulates for achieving optimal pharmacokinetic and/or pharmacodynamic effect, allowing teams to create differentiated and therefore higher value products. The ability to alter the mechanism and rate of release for a drug substance is important. Key single drug products utilizing multiparticulates include Detrol-LA, Adderall-XR, and Agranox. The ability to administer multiple drug substances from a single dose unit is also important. Products with combination or dual release profiles such as Ritalin-LA, Focalin-XR, and Dexilant also employ multiparticulate technologies. I'll point out many ways today that bioavailability enhancing formulations can be coupled with multiparticulates to make these better products. Beyond what can merely be put into the multiparticulates, the reproducibility of gastrointestinal transit relative to monolithic tablets results in more reproducible PK for multiparticulates. Additionally, the spreading out of a full dose across multiple units instead of a monolithic tablet results in lower risk of adverse events from a dose dumping event. Another main target product profile aspect that can be achieved with multiparticulates is the isolation of incompatible drug substances within a dosage unit. This could entail the separation of drug substances from each other in a fixed dose combination, or it could entail separation of the drug substance from water or the functional excipients that are in the multiparticulate to help deliver it. Importantly, drug substance and drug product intermediates are only as good as the dosage form and packaging they are delivered from. Luckily, multiparticulates allow a formulation team the option to use sachets for high-dose situations or patient populations that struggle with swallowing large dosage forms. Multiparticulates are definitely amenable to capsule filling and dosing. Here, the opportunity exists for commercial differentiation and IP protection from the use of increasingly functional capsules, such as those constructed with enteric materials for payload protection and targeted release, or dual chambers for combination drug release. Capsules are also designed for sprinkled delivery to allow ease of use to caregiver and efficacy to the patient that requires it. Finally, tablets are increasingly being used to deliver multiparticulates also. Again, commercial differentiation can arise from the use of multiparticulates in traditional and orally disintegrating or chewable tablets if the multiparticulate properties are designed appropriately. Once the formulation concept is determined, no one wants it to be a stretch or a chore to make that real. We want to scale the process and refine it for robustness of release and yield. Multiparticulate processes are well established in both the fundamental literature and through commercially available equipment. Some of the choices have continuous capability if your team hits on that high-dose formulation or that high-volume blockbuster product. Through today's session, I'll describe how multiparticulates offer a high degree of capability for attaining oral pharmacokinetic, pharmacodynamic, or target product profile goals. In each section, I'll try to highlight the key considerations from the target product profile that led to the multiparticulate technology selection and the designs of each potential product. We'll also touch on some fundamentals of the process that lead to the tunable release that allow attainment of the goals for the case studies. As we design a formulation for any one or multiple drug substances, one absolute must is attaining the desired PK or PD effect. If it doesn't work, it won't matter how elegant or inexpensive a dosage form is to make. Good understanding of the disease state and the target population's compliance is key to a good formulation. Sometimes that means avoiding adverse events such as drug degradation in the stomach. Sometimes we must avoid incomplete absorption, yielding loss of efficacy. To achieve that idealized design, develop, and manufacturing model, delivering quality rapidly, it is ideal to do numerous experiments without committing API or touching stainless steel if possible. This is where pharmacokinetic simulations from a physiologically based model can be invaluable to a formulation team. On this slide, I show immediate release and simple modified release profiles along the left, running through a computational model that leads to pharmacokinetic predictions that are already differentiating these simple formulations in silico. However, we all know that the constraints of the target product profile are rarely so easily attained. The use and mixing of multiparticulates with various mechanisms of release are commonly needed to meet these difficult goals. Many great formulation examples are in the market and academic literature now. Some of these involve higher order profiles or combinations of multiparticulates as shown in the profiles on this slide. One very common problem statement is to require a delayed release of API, as shown in the top right, to improve palatability, avoid gastric degradation, or release drug in the colon for locally acting drugs. Another goal of pulsatile release might be staggered release of 2 APIs for cardiovascular or tuberculosis treatments, or release of a metabolic saturating agent before the efficacious API is released in HIV treatment. Multiparticulates should be a first consideration even if, perhaps especially if, formulation success might require a solubilized or bioavailability enhancing form. A screening event to determine the extent of supersaturation from a simple bioavailability enhancing formulation can be followed by PK modeling and point the path to success before any dosage form is even attempted. Shown in the bottom right is a hypothetical fixed-dose combination with a non-sync dissolution test. This test highlights the dissolution rates, extent of supersaturation, and precipitation kinetics that might result in a worst-case scenario. In that worst case, a well-designed formulation might be able to limit the precipitation by modulating release to match the in vivo absorption rate, thereby limiting the supersaturation in vivo and potential for precipitation. As noted, the primary focus of any formulation team should be on the target product profile components for your drug or drugs, then selecting as simple a combination of multiparticulate and dosage form as possible to achieve those goals. From there, one can harmonize the process and the resulting multiparticulate's physical properties required to reach those goals. This slide is not intended to be exhaustive of all the options available, but it definitely gives an idea of the technologies and process options available. A black and white compare and contrast table would be ideal, but innovation in the use of processing equipment, excipients, and dosage form design makes all of these technologies fairly available for a wide variety of uses. Since companies like Capsugel have all this equipment and know-how ready, this flexibility allows a team to select a technology based on the best mechanism of release or profile for a candidate and disease state. I'll briefly highlight in the multiparticulate types the options that are available, such as pH sensitivity, time-triggered multiparticulates, diffusive mechanisms, and erosion mechanisms of release. Walking farther across the page from mechanism and process, I want to stress that bioavailability, enhancing, or solubilized forms are always an option with multiparticulate formulations. For example, almost any of these technologies on this page can deliver a solubilized form within the low-volume environment of a colon to generate local high concentrations for treatment of local diseases. When a delayed burst coating is difficult to engineer into the multiparticulate itself, innovations such as the intrinsic drug delivery technology involving capsule shells made specifically for enteric protection can greatly speed the pathway to proof of concept. From here, I'll start with case studies, and in each section, I hope to highlight the basics of the target product profile for a candidate or the combination formulation, the selection of a multiparticulate technology for that formulation, and the science and process basics for each multiparticulate type that's selected. The first case study today relates to the compound azithromycin, where there was a need to develop a new high single-dose product to be used by both adults and children to treat community-acquired pneumonia. Azithromycin exhibits high solubility in gastric fluid, which leads to rapid absorption from the small intestine. This can result in high peak plasma concentrations and associated side effects, so there was a need to develop a formulation that provided extended release to attenuate the rate of drug release and absorption. However, as the compound showed poor colonic absorption, a specific release profile needed to be met so that bioavailability was not compromised by the extended release. This product was intended for use in adults and children, so an oral powder for suspension was proposed as the final product concept. Therefore, particles with an acceptable mouthfeel were needed. Another palatability challenge was that azithromycin exhibits a bad taste.
Are you on the phone right now?
Yeah.
Bring it to the phone. Oh, that's fine. Well, there was
Another product requirement was that the convolution of process temperatures and time needed to be minimized to minimize compound degradation. Excipient selections had to be suitable for use in pediatrics, and of course a long shelf life was desired. Based on all of these inputs, the LMP technology was selected as the preferred approach for this product. So, what are lipid multiparticulates, or LMPs? LMPs are round, smooth matrix multiparticulates that are produced from GRAS and or acceptable lipid excipients Most of these are solid at room temperature. After production, the LMP particle size is tunable between 50 to 1200 microns in diameter with a narrow particle size distribution. One of the exciting aspects of LMPs are that they combine some of the benefits of conventional capsule-based lipid formulations, meaning liquid or hard capsules, with the flexibility and additional functionality of a multiparticulate dosage format. Indeed, using an established non-aqueous melt spray congeal process to produce lipid multiparticulates, we are able to formulate a drug with a wide range of different lipidic excipients, including hard fats, waxes, and surfactants. This excipient flexibility and process results in LMPs exhibiting intrinsically acceptable palatability from taste and roughness perspectives as long as the size is tuned appropriately. In addition, it is possible to generate LMPs with sustained or immediate release properties, taste masking potential, or solubility and dissolution enhancement as needed. A product profile might be achieved through a lipid multiparticulate formulation comprising a single excipient or a more complex multi-component system depending on the project and its objectives. In addition, LMPs offer the opportunity to release high compound doses and loadings, while the smooth surface means that they are highly amenable to coating for additional functionality if needed. Lastly, the fact that formulation exists in a multiparticulate form means it can harness all the multiparticulate benefits mentioned earlier, such as good gastrointestinal distribution, swallowability, and the capacity to be used in a range of finished dosage forms. In this slide, I have listed some of the common excipients used in lipid multiparticulate formulations. All of these are GRAS excipients, meaning that this technology can provide formulation options for a variety of market segments, including prescription, over-the-counter, and health and nutrition markets. The core excipients of lipid multiparticulates include hard fats, though additional excipients may be added into the formulation to alter the performance. For example, surfactant may be added to the formulation to introduce self-emulsification properties, which is often desirable when the drug exhibits low aqueous solubility. Meanwhile, waxes or other hydrophobic excipients may offer more sustained drug release rates. An additional point worth mentioning here is that our melt spray congeal process allows us to produce these lipid particles without relying on binders such as lactose or microcrystalline cellulose. This results in higher lipidic concentrations in each particle and therefore greater opportunity for lipidic excipients to modulate or improve performance. The graphic in this slide describes the fundamental LMP formulation properties considering two key inputs, the target drug release rate and the aqueous solubility of the compound. When taking this approach, the fundamental properties of the formulation include the physical form of the compound and the target mechanism of release. For example, when aqueous solubility is high and a fast rate of drug release is desired, as in the upper right corner, the preferred physical form of the compound will be the crystalline form. There's little advantage of solubilizing the compound when a fast rate of release can be achieved via simple diffusion. If taste masking is needed for such a formulation, the LMP may be readily coated or simply dosed from a capsule.
Lastly, in instances where the compound exhibits low aqueous solubility, there are benefits of designing LMP formulations in which the compound exists in the solubilized state. These formulations can contain levels of surfactant and digestible lipidic excipients that recruit endogenous bile salts and phospholipid mixed micelles for effective solubilization and drug transport to the site of drug absorption. Melt spray congeal is a fairly simple appearing process, but there are significant process handles to aid in a formulation team's goals. Starting with an extruder or heated tank, a melt is directed to the key unit, the spinning disc, as drawn and pictured along the bottom of this slide. That centrifugal force generated by the spinning disk accelerates the liquid radially outward to overcome surface tension and viscous forces. That liquid flows and generates droplets that spin off of the disk and are cooling as they reach the sides of the container and conveyed away in the collection cone to further processing. The key process parameter really here is to just have an API property that's greater than 120, melt point that's greater than 120 degrees. At that point, we can probably design a formulation for any of those types of applications described on the prior slide. Ultimately, the shape and size of the disk, the speed and the temperature of the disk are all tunable and used to determine particle size and release rate. The combination of excipient selection and melt spray congeal process handles result in tunable release of crystalline or solubilized API. That release profile can be extended release, as we'll describe for the first azithromycin case study introduced earlier, or a near pulsatile release as described in later case studies and shown in the middle of this slide. For azithromycin, the target product profile components of high dose requirements, a specific release profile for a pH-sensitive drug, and palatability requirements all led us to lipid multiparticulates for this formulation. The formulation is 50% drug with a lipid carrier and dissolution enhancer making up the balance of the formulation. It is processed well below the drug's crystalline melt or dehydration temperatures, so this API remained crystalline in nature throughout the process predominantly. The process results in small, smooth, and fairly monodispersed particles as shown on the right side of this slide. Ultimately, the formulation's lipid carrier was selected not to melt or dissolve readily, yielding a diffusive mechanism of release. Cartoon on the right, we can still envision water coming in, dissolving the drug, and carrying drug solution out. This process continues, essentially drilling down into a particle over time for the aqueous pore diffusion mechanism. This progresses in an orderly fashion as shown in the scanning electron micrographs of LMPs that are embedded in epoxy on the left side of this slide. These have been immersed for the stated times, and the amounts of release from a dissolution experiment are shown in parentheses also. Ultimately, the composition of the multiparticulate is tuned to alter the release rate as shown in the upper left. In this case, increasing the dissolution enhancer and removing the lipidic carrier, more pore former yielded faster release. Because of the drug's pH-dependent solubility, the sachet excipients could also be utilized to alter the release rate as shown in the upper right. Additionally, particle sizes of both API and the lipid multiparticulate have measurable and predictable effects on release rate as shown in the bottom graphs. So through formulation and process, without coatings, one can gain a very good handle on the dissolution rate desired and get a profile that suits the target product profile goals.
A lot of those are really better.
These release rates in vivo, as shown on the last slide, were correlated to areas under the curve and adverse events in human studies, as shown in the bottom left graph. The intersection of acceptability for these two parameters resulted in achieving the target product profile components and yielding a beneficial product for patients. As I've been hinting, lipid multiparticulates and other formulations can be augmented by further coating to attain product profile goals as needed. The next couple studies will rely on fluid bed coating to attain their project goals. As noted earlier, a very common problem in the pharmaceutical market today is taste masking or palatability, which is really more than taste alone. encompassing odor, aftertaste, and texture of the materials also. Most basically, unpleasant taste reduces the palatability of the forms and is directly linked to poor medicine compliance, especially in patient populations such as pediatrics. The risk of taste affecting compliance is increased when treating patients who cannot or do not swallow traditional solid dosage forms such as capsules or tablets. All palatability attributes should therefore be considered when developing a taste-masked or easy-to-swallow formulation. The science of masking taste and common approaches to doing so are highlighted on this next slide. Fundamentally, one can minimize the drug concentration below a threshold through dissolution inhibition to minimize the sensation of taste. One can alter the free drug activity through complexation. One can alter the drug diffusion to the tongue and taste sensors through viscosity modifiers. Or one might be able to alter the perception of taste through flavorants and other additives. Common formulation approaches, therefore, are additives, matrix entrapment, physical barriers, both to release or to absorption, and release modulation based on coating, ion exchange resins, or other formulation applicability. Shown on this slide are basic architectures with the mechanisms of release listed, achievable by fluid bed coating of multiparticulates. We have immediate release layers that can be put onto inert cores, as shown in the left. Those can then be further coated for pH-triggered release, extended or diffusive release, and all of those can use enhanced solubility or dissolution profiles. This cartoon describes amorphous drug, but nano and milled crystals can easily be applied to these types of formulations also. All the cartoons drawn here show inert cores being used, but I'll set up case studies later in this presentation by mentioning lipid multiparticulates and extruded granules can also be readily coated to gain pharmacokinetic or pharmacodynamic effect. The schematic of a very well-precedented process train is shown here. The products with crystalline and amorphous drug layers can be applied through the application of solutions onto inert or active cores. Aqueous coating is commonly more available, but solvent coating can be and remains a powerful tool when it's available to a formulation team to get very good integral layers. There are no limits to the dosage form options if fundamentals are considered into the physical properties of the coatings or the multiparticulates themselves. So key components for an optimized multiparticulate. As part of that compare and contrast for multiparticulates, the elegance of the architectures from fluid bed coating lead to these being an ideal tool for taste masking and other delayed pulse applications. Fluid bed tends to be used for lower loading drug forms, although the ability to use lipid multiparticulates and extrusion spheronization granules as the substrate for coating is reducing that burden. These next two case studies will capitalize on the fluid bed for taste masking and for delayed pulse delivery. This case study relates to the pediatric delivery of a small molecule compound that exhibits high aqueous solubility. It was being explored by a company for the treatment of non-small cell lung cancer in children. Unfortunately, the compound was very bitter tasting. Due to patient compliance issues, simple liquid preparations could not be progressed in the clinic. A pediatric-centric formulation was therefore required, which offered better palatability. It needed to have immediate release properties in order to be bioequivalent to the existing solution and an adult capsule product. Due to the variety of patients in the population, the product needed to be available at many different strengths to enable body surface area dosing via direct to mouth or sprinkle mode of administration. Based on all of these inputs, namely the need for taste masking and immediate release in pediatric subjects, a fluid bed-coated lipid multiparticulate was selected as the preferred technology approach for this product. The developed LMP product consists of GRAS lipid excipients combined with hydrophilic pore formers to provide that immediate release of the drug following administration. However, the lipid multiparticulate was tightly controlled to 150 to 250 micron particle size for optimal mouthfeel, and these smooth lipid multiparticulates were aqueous polymer coated for taste masking protection because of the high compound solubility, moderate to high loading, and the intended mode of sprinkle administration onto food or beverages. The coating level was minimized to ensure that acceptable excipient levels were not exceeded while maintaining immediate drug release profiles. With that in mind, 2 coating levels were progressed into human studies, and the lower coat weight was found to provide acceptable taste masking and palatability while maintaining bioequivalence to existing IR formulations. This formulation is currently with patients and providing a great benefit. This next case study is another application of fluid bed coating to create a delayed pulse delivery and enable a fixed dose combination product. In this case, 2 small molecules with good water solubility were desired to treat behavioral alterations of stimulant abuse. The performance needs included the fixed dose combination with an ideal efficacy defined by one of the drugs being immediate release and a second drug being released 3 hours post-dose of the first drug. As noted, both the modified release drug had a high water solubility and not a large dose. The fluid bed coating to create the delayed burst was a primary option. A multiparticulate format was desired to mix at an appropriate dose into capsules, and obviously appropriate stability was desired by the project team. So multiparticulates based on osmotic bursting mechanisms were targeted here because of the time dependence that was required for this product. Describing the cartoon and reality of this osmotic bursting multiparticulate from the inside out on this page, it's shown as an inert core here because a high dose was not required, and size control was desired since the lag of the delayed burst is altered by the overall particle size, which we'll highlight more on the next slide. Inert cores can also be used for formulations such as this to increase the loading if needed. The drug layer, as shown here, is a crystalline drug form with binder to hold it to the particle, but it could easily be an enabled formulation such as an amorphous dispersion. The swelling layer is the main functional layer combined with the barrier coating layer above it. This swelling layer contains a super disintegrant that absorbs the water, swells to apply a physical force that ruptures the barrier coating above it and release the drug. As noted earlier, the lag time is altered by the overall particle size that's selected. Once that particle size is set, though, the lag is tunable with the barrier coating weight as shown on the left graph. As the barrier coating went from 15 to 18 to 20% weight, with the same drug layer below, the lag time extended from 1 to 2 to 3 hours without significantly altering the release profile once released. For some disease states or fixed dose combination efficacy, such as this one, the timing is vital, independent of where the drug is released in the GI tract. That 3-hour time span for this target product profile was critical and why osmotic bursting was chosen for this fixed dose combo. As shown on the right, it doesn't matter what pH the particle is immersed in. Water activity drives the absorption of water, the swelling of the super disintegrant, and eventually the drug release, independent of the pH and tunable still by the barrier coat weight that's applied. This is even amenable to other types of API drug properties such as size, as long as it's applied smoothly to the particles. I grabbed this literature slide here to show the in vitro in vivo relationship that can be attained by osmotic bursting multiparticulates. As the lag increases in vitro, the lag and Tmax increase in vivo as desired, as shown on this particular case study. So osmotic bursting is generated by fluid bed coating is a powerful formulation option for those indications where timing is of the essence. This next case study is another product enhancement example. As we all know, bioequivalence can be a very difficult goal to achieve without enough formulation tools and handles. Here, a solubilized form was used based on extrusion to generate the multiparticulate for that goal. In this case study, the target product profile included two main objectives: a decrease or elimination of a food effect to help the efficacy and compliance of the schizophrenic patient population that this drug was aimed at. Secondly, the desire was to be bioequivalent to the existing Fed formulation to meet an established therapeutic window balancing the known safety and efficacy of this product. The target product profile is complicated, as always, by a pH-dependent salt form of a weak base being the drug substance, and a moderate dose within a dosage form that was envisioned to release in an IR fashion, but didn't really act that way in real life, as studies during this product development showed. To fix the fasted absorption initially, a solubilized form was utilized. Once the fasted absorption was fixed, though, an immediate release dosage form yielded too high a Cmax for the established safety profile, so modified release granules had to be incorporated into this formulation to meet the safety profile. Extrusion was used here in this case study as a semi-continuous wet granulation concept. In that mode, one can attain very high loadings with a wet massing-like concept. We can formulate poorly soluble drugs with an extrusion concepts, whether that's classical hot melt extrusion to create an amorphous dispersion, or a wet massing of stable forms as performed in this case study with nanocrystals to be described. One can utilize similar excipients for extrusion as in conventional solid oral dosage forms. Here we used excipients to tune the release rates of the granules, but one can also use excipients that would be incorporated for the tableting and other applications that would follow the steps of extrusion. In this case study, we used wet massing and milling. However, spheronization is becoming more commonplace for good reason. It is available in small to large production scales. The extrudates are rapidly and efficiently turned into monodisperse agents by the spinning friction plates, collisions with the walls, and other particles to produce the spherules. And the process parameters are fairly simple to tune in order to come to the desired product profile and particle properties needed. In this particular case study, we preformed the dispersion by spray drying of an amorphous dispersion. We then used temperature and humidity on this supersaturated amorphous dispersion, specifically by design, to generate nanocrystals from the bottom up. These nanocrystals were less than 100 nanometers in domain size, as shown in the TEM images shown next to the cartoon in the bottom right, those sizes being about 50 nanometers in size, or in the powder X-ray that's shown in the upper right, where the broadening of the X-ray peaks indicates a very small domain size in this particular case. As noted, this powder got rid of the fed fasted effect by increasing fasted absorption. However, when put in an IR dosage form, it actually gave too high of a Cmax and too early of a Tmax for bioequivalence. So we started to modify the release rate by extrusion to make bigger particles and mixing with other excipients to modulate that release rate. We extruded and milled in this case, as shown in the upper left SEMs on this particular slide. We also used the composition handle, such as mixing the solid nanocrystalline dispersion with Oidergit L to modulate the release rate by composition alone, as highlighted in the table. Particle size was also a strong handle here for release rate modulation, as shown in the graph in the upper right, where the composite being in the middle versus sieve cuts around it earlier and later show a very strong dependence of particle size on release rate in vitro. These release rate handles, both compositional and process, allowed us to gain control of the release in vivo also. Ultimately, as shown in the bottom, a mix of the spray-dried nanocrystalline powder and granules formulated through the extrusion milling process were compressed into an immediate release tablet to generate a dual release rate dosage form. This resulted in eliminating the food effect and matching the bioequivalence markers of AUC and Cmax for the formulation in both the fed and fasted state. As I start to challenge you, hopefully, with the information from this webinar to reimagine your own formulations, I can easily do that with this particular instance also. In this case, we extruded and milled as shown, but we did not, one might envision that we would have used extrusion spheronization to come to a much more facile process in the future. That leads me to helping everyone hopefully envision how combinations of multiparticulates can help your team succeed with goals. This dosage form for Logimax is a really fine and precise example of a formulation that's very effective and could be reimagined today with different equipment options. To highlight the exceptional nature of this dosage form, it delivers two different drugs that have three orders of magnitude different solubility, but releases them at the same rate. Belodipine is delivered using an oily solution that gets absorbed onto a particulate carrier. This is made by wet granulation in this particular example, but could be extrusion spheronization in the future. The metoprolol succinate multiparticulate is based on a highly soluble drug. It's applied on an inert core by fluid bed in this particular case, but one might envision using extrusion spheronization to come up to a new solid core. The release rate is controlled through a diffusive barrier coating that's applied by fluid bed, and then both of those are compressed into a tablet. Both of these multiparticulates, if generated differently, might have been amenable to a capsule formulation such as a mixed capsule or a dual chamber capsule in the future. So getting back to our idealized design, develop, and manufacturing paradigm for pharmaceuticals, a pharmaceutical team needs the items across the bottom to succeed at achieving speed to market. Capsugel Dosage Form Solutions offers this capability to you through our science and engineering capability to complement the drug delivery capability of the customers, the models, technologies, and equipment to design an optimal and unbiased formulation for any given product, integrated and multidisciplinary sites, personnel on teams, and equipment to increase the speed and decrease the risk associated with any process transfers. And we treat each product as our own and innovate during the entirety of that product to minimize API and time as much as possible. This slide shows the breadth and depth of technologies we discussed today that we routinely use in addressing client problem statements. We address bioavailability challenges via amorphous dispersions, nanocrystals, and lipid-based technologies, whether that's in a drug substance, product intermediate, or in a multiparticulate pertinent to today's talk. We offer a full array of multiparticulate and dosage form equipment and know-how. Hopefully that was in evidence today. These can be combined to suit the full range of specialized applications and product profiles of interest to the pharmaceutical realm. We have these integrated sites with development, clinical, and small-scale commercial manufacturing across the range of solid oral dosage form options. This is inclusive of tablets, powders and capsules, and multiparticulates, both alone and encapsulated. The innovative hard-shell capsule products that Capsugel makes also complements the multiparticulate offering described briefly here today and in other Capsugel webinars. Thanks for taking the time today to join me on this brief multiparticulate tour that I always enjoy giving. If you take only one or two snippets away today, I hope they are from this slide. Multiparticulates are supremely flexible, capable of delivering on multiple target product profile goals simultaneously. The pharmacokinetic, pharmacodynamic, and product profile considerations are and should be the primary drivers in any multiparticulate technology selection. That can include solubility or bioavailability enhancing formulations. Processes for multiparticulates are fundamentally understood and commercially available, and that includes drug layering from fluid bed, extrusion, and spheronization. I didn't touch on mini tablets today, but that's definitely a multiparticulate option, and melt spray congeal was highlighted earlier. Capsugel dosage form solutions would be happy to offer these technologies and integrated capabilities to help you deliver solutions from design through commercial manufacturing. With that, I thank you again, and I'll now start to field questions from the audience.
Thank you, Brett. Now let's move on to the Q&A. There's still time to submit your questions using the Q&A box at the right side of your window. We have lots of great questions already, and we'll try to get to as many as possible. The first question is, what is the difference between spray drying and fluid bed drying? What is the advantage and disadvantage of each?
It's a good question. Thank you. Both of these technologies, spray drying and fluid bed drying, are atomization techniques to take a droplet and turn it into a powder or a solid form. But they are employed in very different ways. Spray drying, usually you're not coating any particular substrate, but turning that into a powder in and of itself that can be further incorporated into any dosage form. Fluid bed coating is you can atomize the same aqueous or solvent-based solution and turn that onto a substrate or use it to granulate substrates that are inside. So to compare and contrast, one can end up with the very same physical state, meaning an amorphous or a nanocrystal embedded in a material. And those materials can be further encapsulated or tableted as necessary. So it gives you two different methods by which to either come to a powder or a particle that can be used for dosage form effect.
Thank you. Next is, what does a typical design, develop, CTM manufacture campaign look like for API use and timing?
That typically starts for us with designing a formulation based on the drug properties with our client hand in hand, since they know the drug better than anyone in the world at that moment in time. Usually we like to work in development cycles of roughly 2 to 3 weeks in time, whether it's powders or particles or tablets that we're determining. Usually it would take 1 to 2 cycles perhaps to lock in with the models on concepts that suit the problem statements of the target product profile. We'd commonly provide some animal study supplies from the later cycles of that, perhaps within 2 to 3 months. And as success is found by the client, rapidly move through method development and validation into clinical manufacturing beyond that. So commonly we'd start from a kickoff design to clinical manufacture in a 6-month time span, depending on the client need. And commercial manufacture hopefully is the outcome of all of that, and that would just be dependent on the project and its clinical needs.
Thank you. Next is, can you comment on the drug loading range with MP approach?
So drug loadings are somewhat amenable and somewhat dependent on particle properties such as the API if we're going to make a particle with crystalline drug or an amorphous drug entity. As I noted throughout, hopefully, fluid bed layered multiparticulates, since they commonly start with inert cores, usually are used for lower dose or lower drug-loaded products. However, the use of more active cores from extrusion spheronization or lipid multiparticulates can increase those uses, and so that's where lipid multiparticulates, any extruded or spheronized granule, or even a mini tablet can usually have a higher drug loading, and coatings can be applied in a minimalistic way, hopefully, to attain the need of the project.
Thank you. Next is, what loading can be achieved with LMP technology or osmotic burst technology?
I'd say commonly with a lipid multiparticulate, in a crystalline drug application, we commonly use it in the 20 to 50% active range, and that's somewhat dependent on the API and somewhat dependent on the particle size of the API. So there's a little bit of tuning that usually goes on early to find the right melt, to find the right process by which we can get a nice viscosity and temperature profile from the disc that will allow for efficient atomization of the material. Osmotic bursted multiparticulates are commonly a little bit lower in loading, again, from the fluid bed application, relying on inert cores commonly.
Thank you. This will be our final question. We had a lot of great questions today and couldn't get to them all, but we will be getting back to everyone who submitted personally after the webinar. The question is, are there any limits to the final dosage form that can be utilized with LMP or osmotic bursting MPs?
I can fairly confidently say that the answer to that is no within reason. There's always, whenever you're making a multiparticulate, capsules are obvious for both adult or pediatrics through the sprinkle capsules that allow caregivers extreme flexibility in delivering a dose to a drink or a food. The coatings that get applied to osmotic bursting specifically, and those particles, usually as long as the multiparticulate is designed well, commonly that means it has a fairly small size and a tunable materials around it in the immediate release tablet. It's fairly readily amenable to tableting also. So usually with LMPs or coated multiparticulates, we can figure out how to get them into capsules, tablets, whether that's a conventional IR tablet or even an orally disintegrating tablet, and sachets are obviously available also.
Thank you, Brett. And we have the final poll question, which is, would you like Capsugel to follow up with you after the webinar? Thank you for attending this Fierce Live webinar and submitting so many great questions. I'd like to thank our speaker for participating and Capsugel for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours on the same page you used to register for this event. Thank you again for joining, and we look forward to seeing you at future events.